we
the...
said piecewise.
Oh, that too.
And
Okay.
he said he really likes
He
the
probably
different
just
colors.
Chapter 8, photosynthesis. So today I'm covering the first 3 sections of photosynthesis, which will give us an overview. And it's going to talk about the history of photosynthesis. Like, how did we know that it's made up of light-dependent, light-independent reactions? How did people know what plants were taken in to undergo the process of photosynthesis? And then the last section deals with pigments like chlorophyll and carotenoids. So, I guess my mouse is in here too.
Ideal.
Okay, so a little bit of an introduction. Almost every oxygen molecule that we breathe was once part of a water molecule liberated by the process of photosynthesis. All energy, you can trace it back to the sun, but essentially the food that you eat to get energy is directly or indirectly by photosynthesis. So without photosynthesis, we wouldn't be able to utilize energy from the sun. So I think, in my opinion, photosynthesis is one of the most important processes that has ever occurred in order for life to take off. So a little bit of an overview. It combines essentially carbon dioxide and water, producing glucose and oxygen. And there are a few different ways to undergo photosynthesis. You can have anoxic organisms, which means that they don't produce oxygen. So some species that undergo photosynthesis and don't produce oxygen as a byproduct, purple bacteria, green sulfur bacteria, green non-sulfur bacteria, and then heliobacteria. So there's some pictures here.
James just Snapchatted me and said, "Can you tell her to zoom in?"
I can't see you. I can't see anything. So that means they can hear me and see me, but I can't see them.
That's whack.
What's he saying?
How
And
can
he
they
said you guys are
possibly-
frozen.
How can they possibly hear you?
He said we're frozen.
So there's no signal there.
It's got to be on their end.
Yeah.
I've restarted this at least 7 times.
Really?
So, yeah. Okay, so then oxygenic means that they do produce oxygen as a byproduct. So, yay, cyanobacteria, probably responsible for the oxygen levels we breathe today. The 7 different groups of algae that can undergo oxygen-genetic photosynthesis, and essentially all land plants. Okay, so photosynthesis, where it occurs in the leaves, but more specifically in the organelle, the chloroplast. And there's 3 stages. Light-dependent reactions, which means that it depends on light. So we'll get energy from the sunlight. We'll talk about how cells do that. And then using this energy to make some ATP, and then reduce, oh man, look at this molecule, NADP+, which is an electron carrier. So it will pick up electrons and become NADPH. So very similar to our NAD+, NADH. It's just that this electron carrier is found in plants. And then, so this is kind of the first 2 stages. And then the 3rd stage is using that ATP as well as that electron carrier, H, to power the synthesis of organic molecules from CO2 into the air. And so this process can occur with light or without, okay? But, so this is the light-dependent reaction. This is the light-independent reaction. A lot of times we call light-independent reactions carbon fixation because it's taking in CO2, and then that's where we'll cleave off some carbons and rearrange it to form our carbohydrate glucose.
Still think that the person who decided to name gaining, reduce, losing.
Yeah. So here's the simple equation for photosynthesis, which you do need to know for your exam, which some of you did not do very well with cellular respiration. So please, this is a point on your exam. It's not a very hard equation. 6 CO2 plus 6 molecules of water yields that sugar molecule, C6H12O6, and then 6 molecules of oxygen.
So it's basically the opposite.
It is the opposite process. Okay?
You just don't need enough energy.
Right. Okay, so it takes place in the chloroplast. So we're going to talk about the structure of chloroplast, the thylakoid membrane. So an internal membrane of the chloroplast, continuous phospholipid bilayer organized into flattened sacs. I do have a diagram on the next slide that shows these structures. And then this is where you'll find the chlorophyll, the pigment that captures light energy. The chlorophyll is rearranged in photosystems, and so we'll talk about the two different photosystems of a plant. Stacks of thylakoid produce grana, so you'll see that on the next slide here. So here's my plant cell, very boxy-like with the cell membrane. The green organelles, those are the chloroplasts because they hold chlorophyll pigments. Here's a bunch of thylakoids stacked together to produce grana.
So thylakoids like the pancakes.
Yep.
Thylakoids.
Okay, here's a real-life picture. So this is 2D light microscope. Yes, and a cartoon showing the two here. But chloroplast, again, you can see the stacks of thylakoids producing your grana. So lots of surface area, and we'll talk about why this chapter. Okay, so another structure inside the chloroplast, the stroma, a semi-liquid substance that surrounds the thylakoid membrane. And inside the stroma, we have lots of enzymes that can undergo chemical reactions. It's basically taking CO2 and using energy from ATP to reduce NADPH. We'll also find the photosystems. So this is where your pigments hang out, and they kind of rearrange themselves kind of like a satellite dish. I like to think of it as that, where light comes in and it bounces it to the center, and then that's where kind of the magic happens with capturing light energy. So light with the proper wavelength, and we'll talk about pigments here, strikes a pigment molecule, the electron is excited, starts to pass energy to another pigment molecule. There's no physical transfer, they're just transferring energy. It arrives at a key chlorophyll molecule, which transfers this excited electron onto the next kind of protein channel, resulting in ATP and NADPH. So here's photosystems. Hold on, I just got a text from James. We have nothing, it's not working, Tracy's calling the company.
Oh, they're on their end?
Yep.
Okay, so it is theirs.
All right.
All right, thank you.
I'm recording. Just have to watch it.
This is why combining classes.
Okay, so there are two photosystems in the chloroplast, and it's named weird. So notice it says photosystem 2, and then we have photosystem 1. Well, first they discovered this photosystem, and then they realized that it was working in tandem with another photosystem. So they named it in the order that they discovered it. So yay, awesome. So this is it goes photosystem 2 first, and then photosystem 1. So essentially what happens is I have this antenna-like complex that has these pigments that capture light energy, or in this case a photon. And it bounces around to a reaction center, and this electron gets super excited, where to the point it gets picked up by electron carriers like plastoquinone, abbreviated Q, okay? And it travels through some protein carriers, just like an ETC, but this one is named B6F complex. And as it's traveling through these protein channels or carriers, hydrogen ions are getting pumped across, just like in cellular respiration in the ETC. Then it gets picked up by another carrier, plastocyanin, abbreviated PC in this diagram, to the second photosystem, in this case photosystem 1. Bounces around, gets excited again, gets up to a higher level of energy, gets picked up by ferredoxin, and then this is where the electron carrier picks up that electron with the NADP reductase enzyme, forming your NADPH, which will be used in a different process in photosynthesis. So yeah, you have to know this diagram. It doesn't look like this. It looks like the one in your book. Okay, and I don't know what figure that is offhand, but you do need to know the photosystem. So just find the one that looks like this. Like a Z. Yep, so on Thursday, you'll be quizzed on photosystems 1 and 2, and then on Friday, the Kelvin cycle.
Do you have the book like downloaded?
Oh, you know what? Hold on. Not yet. I guess I can. I'll have to do it when I'm done here.
Yeah.
Okay. Now, before I move on, where did the electrons come from?
Sunlight.
That's where the energy comes from.
The water.
The water. Okay. So you notice that plants take in water, and then this energy actually splits water into protons, which go here, releases oxygen, which we breathe in, and the electrons are the ones that get excited and moved and carried on to pump those hydrogen ions across. Okay, so how in the world did we ever discover photosynthesis? It starts with a guy, I think his name is Jean actually, Baptista von Helmont, a Belgian doctor in the 1600s. And what he did is he planted a really small tree in a pot of soil, and he weighed it, the tree and the soil before. The tree was only like 5 kilograms at the start of the experiment. And then he weighed the soil. He grew the tree in the pot for 5 years, and the only thing he added was water. The weight of the tree at the end of his experiment was 74.4 kilograms, so it pretty much added 70 kilograms of weight. But the soil in the pot weighed only 57 grams less. Yes, 57 grams less, so only a pinch lower than where we started at.
So, the soil and why is the pot constant?
Yeah, it's a constant. But
They're saying there was just a little bit less soil.
yep, just a little bit less soil. Maybe when he watered it, some soil spilled over. Well, maybe it was windy. I don't know where he kept the pot, so whatever. Okay. So he concluded that the weights of the tree, where did the tree get this extra weight? He incorrectly concluded that water accounted for the increased mass, because back then people thought that mass came from the soil. Like trees and plants took things from the soil and used it to build and grow big and gain mass. But he said, nope, it's not the soil. It's got to be the water. It's the only thing I added to it. But however, he was incorrect. So we all know that plants need soil, light, water, and air. And so what are plants made up of? Well, people thought soil. So do plants really eat dirt? He went out to solve that. So here's this nice little von Helmont little tree experiment. 5-pound plant, 200 pounds of soil 5 years later. Nope, it's not soil. So next came Joseph Priestley, an English scientist, 1700s to the 1800s. What he did is he took a piece of mint, okay, and he put it into air with a wax candle, and it burnt out on a certain day. Then he found another candle that could be burned in the same air, August 27th, and then somehow he realized that the sprig of mint restored the air so the candle could burn, because we all know that fire needs oxygen. Okay, so I know this probably doesn't make sense when bullets, but the picture on the next slide will. So then he decided to test to see if a mouse could restore the air. Well, the mouse suffocated and died. So living vegetation adds something to the air. That was his conclusion. So here's a diagram that does a way better job with my bullets. So candle in like a glass vial. Light it, cover it. After a short time, it burns out because it burned up all the oxygen. So then you put a sprig of mint, and the candle continued to burn for days. So then he's like, all right, let's see if the mouse does something. Nope. Okay. Now if I combine all three, sprig of mint, mouse, hey. So it's got to be the plant. Plant's doing something to the air. It's adding something back.
The mouse was probably breathing in that water. Using up the air.
So how does vegetation restore air? Jan Ingden House, Dutch physician, 1700s, demonstrated air was restored only in the presence of sunlight and only by plants' green leaves, not the roots. So they knew that the plant added something back, but then exactly what part of the plant. So he proposed that green plants use sunlight to split CO2 into carbon and oxygen, and that this oxygen that was released combined to form carbohydrates, the sugars that plants make. And so by the end of the 19th century, they determined that the overall reaction for photosynthesis was CO2, water, light energy, because you need
Could a custodian contact the office, please? A custodian contact the office?
forms a carbohydrate of some sort, okay, and then oxygen because of the candle stayed lit. So here's another experiment with light and dark. So we have the plant and the candle, okay? And I don't know, I think it is supposed to be lit. But in the presence of light, the candle is lit. You can tell that one's lit. I just think the picture is just too pixely. And then in dark, the candle went out. And then he's like, okay, what about a potato? There's no green plant or vegetation. It's starch. It's just stayed unlit, basically, or died out. So green plants do something with the light. So photosynthesis includes both light-dependent and light-independent reactions. So moving on in our little history of photosynthesis here to Blackman. So late 1800s to early 1900s concluded that photosynthesis is a multi-stage process with one portion that uses light directly. And so he decided to measure the effects of different light intensities as well as carbon dioxide concentrations and temperatures on photosynthesis. And this is what he came up with. With low light intensity, he increased the light but not the temperature or the carbon dioxide concentration, photosynthesis increased. With high light intensity, increased temperature or carbon dioxide concentration, photosynthesis increased. So he started to mess around with variables and notice different rates of photosynthesis. He concluded that the set of reactions depend on light and that a second set, he called dark reactions, happened, but it wasn't light dependent. He also found that if you increase temperature to a certain rate, it increases, but then after 35 degrees Celsius, it tapers off. And that's because we know that if you alter protein enzymes, they don't work right anymore.
It would be so frustrating to be alive back then because you just have to go by the explanation that, yeah, this green plant does something.
Mm-hmm.
Oh,
Well, I was just thinking about it this morning, you know, how like, oh, we can't do labs because we don't have equipment. And then I think about these guys and I'm like, no, we could do stuff because like they didn't really have anything. I think sometimes, I won't say it, but you know, he doesn't do as many labs because we don't have all this equipment. But it's like, yes, you can. You can do other things, just not the cool stuff.
you can
I don't know.
wing it.
You can wing it, yeah. Okay, so here's a graph showing the rate of photosynthesis and light intensity. So if you increase the light intensity, eventually it will taper off depending on what you mess around with. So in this case, here's 20 degrees Celsius with temperature, it increases and it will taper off. Here's 35, tapers off. Insufficient CO2, 20, this is what you're going to get. So all thanks to Blackman. All right. Now, people thought that the oxygen the plant gives off comes from CO2, that they cleaved the carbon. So this is the oxygen that gets released, and then this carbon is used to make sugar, but that's not true. The oxygen that we breathe in comes from the splitting of water, which you saw in the photosystems. So when water splits, that oxygen is what we breathe in. That's the product, okay? CO2 is used in sugar formation.
So I mean, we're like halfway from being able to breathe underwater. We just gotta find a way to get rid of that.
Find a way to split water as you breathe it in. Yeah. That'd be kind of a cool device, actually.
We just gotta get rid of the hydrogen.
That would be an awesome device. Okay.
We should make one of them.
C.B. Van Niel studied purple sulfur bacteria. So this PSB is the purple sulfur bacteria abbreviation. So these guys don't release oxygen in photosynthesis. Instead, they take hydrogen sulfide and they turn it into pure sulfur. So this is the reaction for photosynthesis in the purple sulfur bacteria. You take CO2, hydrogen sulfide, light energy, you still make your carb, your glucose, water, and then sulfur is released. So he proposed this generalized equation where A, it could be sulfur or it could be oxygen. Okay, so CO2 plus 2H2, whatever you're looking at, oxygen, sulfur, there's different forms of photosynthesis out there. Makes your carb, your water, and then whatever. So, molecule of oxygen or sulfur. So it serves as an electron donor. That's where the A is. So in photosynthesis, the electron donor is the water or the oxygen. Product A comes from the splitting of it before. Therefore, oxygen produced by green plants results from splitting water, not CO2. Now this was an amazing idea. But now you got to go out and you got to prove it. So they actually had the technology back then to prove it. So the oxygen released by photosynthesis comes from water rather than CO2. So here's the experiment. Give plants an isotope label of water and unlabeled CO2. And then in the second experiment, give plants isotope labeled CO2 and unlabeled water. So going back over here, labeled water, they noticed that the oxygen released is labeled because the water was labeled, so when the H splits and the O splits, they could like trace it and say, yeah, that's where it came from because we tagged water in the oxygen that was released. We saw it labeled fluorescent dye or whatever, I'm not exactly sure how they dyed it, but there are ways to do that. In experiment 2, they labeled the CO2, and then what was released was not labeled. It stayed in the plant as sugar/starch. So it's kind of cool. All right, so ATP and NADPH are made in light-dependent reactions, and they reduce CO2 to make that glucose. So then Neal went on even further to propose that the H ions and electrons generated by the splitting of water were used to convert CO2 into this organic matter called carbon fixation. So again, big contributor to photosynthesis. 1950s, Robin Hill proved that Van Niel was correct, and he demonstrated that light energy could be harvested and used in reduction reactions. What he did is he went out and he isolated all the individual chloroplasts of leaf cells and just like studied it, figured out what's released, where does it come out of the plants and all that. But there's more. There's a bunch of more experiments past 1950s prove that electrons released from water splitting were transferred to this electron carrier NADPH. And so if chloroplasts are deprived of CO2, they accumulate ATP because ATP is used in the process of carbon fixation. So if CO2 is introduced, then ATP and NADPH accumulates, and then it's assimilated into the organic molecules because these guys are used to make sugars. So who cares about these experiments? Well, from these experiments, we have concluded that photosynthesis happens in plants within the chloroplast. It showed that light-dependent reactions use light energy to have NADPH reduce, which means it gains an electron, and it becomes what? Yep, NADPH, okay? And to make ATP, and then these two molecules, well, the reduced form of it, are used to form simple sugars with the intake of carbon dioxide.
How do you measure the number of electrons? Is it like the same number as the number of protons?
Yes. Usually, you assume, unless there's a charge.
That's why it's neutral. So that's why, so each one of those NADP plus is just carrying one electron.
Yes. Yep. Okay, the last section is pigments. So how they absorb energy from sunlight. So pigments, molecules absorb light energy in the visible range. The color we see is a color that is not absorbed. It is the color that is reflected. So like a red apple absorbs all the colors except for the red spectrum, and that hits your eye. If you're wearing a black shirt, okay, that means that it absorbs all the colors and nothing's being reflected to your eye. If it's a white shirt, all the colors are being reflected to your eye because white light is made up of the rainbow, right? So it's kind of hard to grasp. Are there any questions on that?
Wait, so black
I
light
always
is
thought-
none? Or all of it being, like, what was the black?
Okay, white light is made up of Roy G. Biv, right?
Yeah.
So if you see something white, that means you're seeing
All the colors.
all the colors. A black sweatshirt absorbs all colors and nothing's being reflected to your eye. That's why like dark is dark because there's no light.
Alright, that's
Isn't that crazy?
I was thinking that the black was all the colors mixed.
That's paint. Light is different.
You should get a can of Vanta Black or whatever.
What's that do?
Oh yeah,
Black is black.
Yeah, it's like
Yeah.
so black it looks like there's just.
It looks
like
fake.
nothing.
It looks like it's Photoshop.
Yeah,
Yeah.
everybody looks.
Oh,
It's like
really?
yeah, they
Every
paint
picture
it. It
you see
looks
looks
like it's just fake.
Photoshopped.
Look at they painted this head. Oh, we already got it up.
What? That
Let me
is
see.
crazy.
Well, like you can't tell like that.
Just like a black hole.
All right, so light is a form of energy. Understand that light behaves as waves and particles. So there are wave natures of light. That means they have different wavelengths and different frequencies. Visible light is only a small part of the electromagnetic spectrum, and it's the only part that you can see with your eye. So you can divide visible light with a prism. So we talked a little bit about that on the previous slide. Light is also a photon, a particle of light that acts like a discrete bundle of energy. So it acts like a wave, but then again, the particle nature of it. So like photons can actually like move things. They act like a particle and they behave like light. Einstein is the one that figured that one out. So here's the electromagnetic spectrum listed from lowest energy to highest. And then here's the light spectrum. And if you've ever wondered why we call it infrared, infra means below the red spectrum. And then ultraviolet means above the violet spectrum. So if you're wondering why they called it infrared and ultraviolet, that's why. It's kind of cool. So lowest to highest. And then I know this picture's upside down, but I correlated it to the table. So these guys have higher energy, but their wavelengths are smaller. Okay? These guys have lower energy, but their wavelengths are bigger. So I mean, like the wavelength of radio waves can be 100 meters to like 100 feet. I mean, they're huge. The length of a football field. Okay. Diagram of a photon. Photon's a piece of energy, has no mass, moves at the speed of light, acts like a particle, does have momentum because, again, because it can move things, and it can be quantized by like little arrows, like packets. And then a little joke here. A photon checks into a hotel and is asked if he needs any help with his luggage. No, I'm traveling light.
Woo!
Okay, wait, so you know how like force is weight times mass?
Yep, it's a mass times acceleration.
So if you had just a tiny object, like a speck of grain, it hit you going at the speed of light.
Would it go through you?
Well, that, or would it like knock you backward? Because there's so much speed.
You know what? I don't know, because this kind of reminds me of the Mythbusters with the penny from the top of an Empire State
Yeah.
Building.
Oh, and how it could like kill you or something?
Yeah, but they busted that, but that wasn't moving at the speed of light. And then they shot each other with guns. That was always good.
That's probably
That seems
I feel like it would definitely go through you just because it would have momentum.
like the speed of light, like that'd be so.
I mean, definitely, because I mean, like, think about playing cards in a tornado. You guys have seen pictures of like playing cards on pieces of paper
Or
in
like wheat.
Yeah.
embedded.
They get like stuck inside a.
Yeah.
So I think anything moving at the speed of light would mess you up. Except for the except for the little part of it.
Maybe like
Except for the photon, because it obviously has. I don't know. This is just blowing my mind right now. Okay. The energy content of a photon is inversely proportional to the wavelength of light. So I kind of showed you that diagram. Short wavelength, higher energy. Long wavelength, lower energy. There's this really cool effect called the photoelectric effect, and this is discovered by Einstein back in 1905, when a beam of light is able to remove electrons from certain molecules and it creates an electric current. And they notice that this happens with metals. So short wavelengths, because they have higher energy, are more effective to strip electrons and create electricity, because electricity is just basically a flow of electrons. So here's the diagram that depicts this photoelectric effect. Here I have this metal, in this case potassium, and in order to move electrons from potassium and to generate electricity, you need 2 units of energy, or 2, I don't know, EVs. Well, red light only has 1.8, so red light spectrum, no electrons are being released. Green light, 2.3, it meets the threshold, but lower energy electrons. Blue light, 3.1, super high, high energy electrons being stripped and to generate electricity. So chloroplasts are actually tiny little photoelectric devices that absorb sunlight and then electrons being given off and released at higher energy levels to
Just like
this is electric area.
the other day in Rockford, I thought there's like different things are different heights though. So
Are you guys on electric configurations?
Yeah. Yeah.
Yeah, I'm teaching my kids that right now too. Yeah. And you guys memorizing the 1s2, 2s2,
Oh my god, I hate that.
2p6, 3s, and then all of a sudden you're just like, wait, why does 3d
Wait till you
come
guys
before
get to acids
4p?
and bases.
Yeah.
Oh, it's bad.
Okay, so each pigment has a characteristic absorption spectrum. So each pigment only absorbs a certain wavelength spectrum of light, we'll say. Photon strikes the molecule, energy is either lost as heat or absorbed by electrons. And then they get boosted to higher energy levels and travel through the photosystems. A Absorption does depend on how much energy it carries, the wavelength of it, and then the chemical nature that the molecule hits. So this diagram, oh, never mind, it must be on the next slide, my bad. I'm excited here. So we call this the absorption spectrum. And there's basically two pigments in plants that absorb energy. We have your chlorophylls, which are green, okay, and then your carotenoids, which are like the orange-yellow. So two general types are used in green plants. Yes, you can find chlorophylls and carotenoids in both. So in this little flow chart here, with photosynthetic pigments, either chlorophyll or carotenoids, and there's even classes underneath it. So we'll talk about chlorophylls A, chlorophylls B, and then the carotenes and the xanthophylls. So chlorophylls have a narrow range of photons. There's two types, chlorophyll A and chlorophyll B. Chlorophyll A is your violet-blue spectrum light. It is the main photosynthetic pigment that's found in plants and cyanobacteria, and it's the only pigment that can act directly to convert light energy into a chemical energy. The other ones have to like funnel together or do something special to do that. Chlorophyll B, they absorb the red lights. We call them accessory pigments or secondary light absorbent pigments, and they just complement and add energy to chlorophyll A. Neither absorb wavelengths between 500 to 600 nanometers, and this is why we perceive plants as green, because this is the green light that's being reflected to your eye. So that's cool. This diagram does a nice job, I think, showing you the absorption spectrums of chlorophyll A, where it's the blue, chlorophyll B, which is this shade of blue, but nothing for green really. We haven't talked about the carotenoids yet.
What about that?
Oh, and here's chlorophyll B with the red, just to see accessory pigments.
What about the 5-collier?
Yep, that's also a carotenoid.
That bad boy has a little bit of
Okay, the structure of chlorophylls. So because they can absorb photons and they can excite electrons, this is basically the photoelectric effect going on here. And then when they started to analyze the structure of chlorophylls, they noticed that the shape of chlorophylls is very similar to hemoglobin, which is cool. So what I mean by that is that both hemoglobin and chlorophylls have like a ring-like shape. In chlorophylls, we call it the Porphyrian ring, and it alternates single bonds. But in the center is a magnesium atom, which is a metal, which can, where electrons can be excited because of it. So when this happens, we usually call it the action spectrum. So here's this porphyrin-like ring structure, and there's magnesium smack dab in the middle. Here's hemoglobin, iron's in the middle. Here's plant chlorophyll, it's just magnesium, but very similar shape. I think there's one more difference. Here's OH, here we have string carbons. OH, carbon, double bond. Yeah, but there's very, very few differences between the two. Okay, moving on to carotenoids and other accessory pigments. So, carotene, I can't even say that. Xanthanaxifen, I don't know. Lycopene, you may have came across that, I think, in ketchup. So these carotenoids, they are accessory pigments. They absorb photons with a wide range of energies, but they're not highly efficient as the chlorophyll A. But they do assist in photosynthesis by capturing energy from sunlight. Carotenoids play a different, more important role, I think, in free radicals. So maybe you've heard of radicals, maybe you haven't, but in redox reactions, chloroplasts generate destructive free radicals. And these radicals go out and they can change the DNA in cells. They can, for us, it can cause like cancer. That's why we take antioxidants, or hopefully you do. So carotenoids act like antioxidants to lessen the damage. It's a protective role. And how it does that is it splits two molecules of vitamin A and can make radicals less harmful to the plant. So because two molecules get split and you form vitamin A, this is why carotenoids are really good for your eye vision, because you use vitamin A for vision. So here's a diagram that shows...
So it's actually like, so does eating carrots actually make you see better, or is it
No, it's because it has vitamin A.
But like how much does it actually?
I don't know how much. I just know that...
Like, is it worth it to eat
Yes.
carrots?
Okay, maybe not a bunch every day, but getting your carotenoids weekly, yes. So, like, remember... Oh, maybe I haven't talked about this with you guys. Did we talk about genetic genetically modified organisms and golden rice. Do you remember golden rice at all?
I kind of feel like I barely remember, but
Okay, so like in Africa, they don't have a supply of vitamin A really from their crops that they grow. So then in the 1950s, they took rice and they took like the gene to make carotenoids / vitamin A and they inserted it into rice, and it's called golden rice. And these people, before golden rice was invented, had eye problems, glaucoma, they went blind, and then they introduced golden rice and all those problems disappeared. So yeah, it's...
so if a blind person ate a ton of carrots.
Okay, no, you can't reverse really, but you could prevent. Okay, so this does we call this the antenna complex with all the pigments, and it starts with like the ones that really don't do much, and then it filters to the ones that do the most. But carotenoids to chlorophyll B to chlorophyll A to that reaction center where electrons get excited. Here's an antioxidant. Here's a free radical, and what antioxidants do is they transfer extra electrons to the free radical to neutralize the effect. So, skull, blue, happy. I don't know. Okay. So here's a stable molecule. This is an unstable molecule. So we just make molecules stable. So that's part 1 of photosynthesis.